gene tree. MSA and SNP analysis generates a multiple sequence alignment, and you could use the multiple sequence alignment in the gene tree tool. So inputs to the gene tree tool, you can specify right at the outset whether you're interested in doing DNA or protein analysis. I'm clicking protein. You can select a FASTA file from your workspace and it will populate this drop-down table. Since I've selected proteins, it just gives me only the FASTA files that are of type protein, because we tag our FASTA files by whether they are nucleotide or protein. So you can have your own FASTA file, which you could have uploaded and gotten from anywhere. This is also further detailed. This is already aligned FASTA, so it would skip the alignment process and just build the tree from it. Having done the alignment in the previous tool, we could have gone to that actual alignment and used that here. The next one is unaligned FASTA. So this is sequences you've uploaded into a file in your workspace, and it would align them and then run the tree building on that. Feature group is another way of specifying your input sequences. That's what I'm going to choose right now. To do this, that's the drop-down view. It quickly looks at feature groups that you have in your workspace, but I'm going to click on the folder icon to the right, which gives you a little more ability to search through the spaces. Click on public workspaces, scroll down past all of those redundant names at the top to BV-BRC workshop, gene tree folder, and here is the exact same feature group that we just used for the MSA example. But I'm going to click OK on that. And since this is a feature group, this is the list of genes that I want to align. This DNA versus protein radio button at the top tells you whether to pay attention to the DNA or the protein version of those genes. We're doing proteins. And just for completeness sake, this next possible entry is for a genome group of viruses, and you could build a tree from the whole genome alignment of a set of viruses, as long as they're 250 base pairs or shorter and single segmented. As in so many tools that allow you to accumulate multiple inputs, you have to push the plus button to move a specified feature group down into the box below. And that box below could accumulate multiple entries. So you could have unaligned FASTA plus this feature group, and it would align them together and then build the tree. Over here on the right side, there's some parameters for doing the job. And here is where I mentioned that ragged end on the beginning of the alignment. Trimming off this big gappy area would just be, I think, a good thing to do for tree building. And this control here lets you specify how many gaps you're willing to tolerate at the beginning of your alignment or the end of the alignment. I just go with the value of 0.5. So it's going to trim until at least half of the rows in the alignment have nucleotide data in there. So on this alignment, I think it would probably trim to about somewhere in here, maybe 14, 15, or 16 base pairs. Then this other control, I'm going to set it at half. It's going to remove entire sequences. You'll lose sequences out of your alignment if they are more than whatever this proportion is of gaps. So if there's a sequence that's more than 50% gaps after the first trimming, it's going to be kicked out too, which can frequently be a good thing if you have some things that just don't belong in there. It will clean up your alignment quite a bit. Below that, for tree building, we have 3 different tree building programs: RaxML, or PhiML, or FastTree. And my only guidance here is I like RaxML. FastTree would be preferable if you had a large number of sequences. Like I would say, if you had 100 or more, FastTree would have a much more reasonable runtime than RaxML. RaxML might go for an hour or hours. But since we only have 30 or so in this genome group, RaxML will not be a problem. And then for the model, this is the substitution model for protein statistics for calculating the maximum likelihood tree. LG turns out to usually be the best model for most bacterial proteins, in my experience. But there's a set of others that the dropdown will let you select. Okay, I'll just put it there, and endosymbiont bioredox tree. I'm just going to submit this. But we don't have to wait for it to actually run because this might take a while. And the reason is just because it's on the queue. To actually run the job will be pretty quick, but it sits in the queue for a while because I think with this workshop going on and other work that the computers are being kept busy these days. I have the job pre-calculated, so I'm just going to go back to this workshop directory. Here's our protein tree. This other version is the nucleotides. Within the job output folder, we have the eyeball viewer. That was launching the Archaeopteryx tree viewer. And that little error is just a very small error. If you click the reload button, it'll fix it. Here's the tree, and the tree looks good as far as the tree part of it, 100% support on most of the branches. The identifiers are not really that useful because they're very long and they are just database identifiers for the genes. They don't tell you the species and the strains. I have to apologize because I thought that we had gotten this working such that you had choices on the left side. For example, you can control node name to turn it off. There's supposed to be species, strain, and some other options selected from the database that you could use to label the tips on this tree that are not currently active, but they should be made available really soon. But I just wanted to point out that these controls on the left of the Archaeopteryx viewer are the ones that you would use to turn on and off different ones. So here's an example of turning them off, but we don't have other ones to turn on. Here's an example of internal labels. You could turn off those support values. That's not the only view of the tree though. Go back. We do have in the HTML report, same view of the tree exactly with the same exact labels. Here are details about the submission of the job command line that was run on the job. Yeah, there's something definitely not hooked up right, because here it is reporting that it was writing a Philo XML file with these database items. So just something is not hooked up right in terms of enabling those fields to be viewed within the Philo XML file. Yeah, sorry about that. All of these different views only have those gene identifiers, and that's not generally useful. I've got to solve this problem for you. We go back to the HTML thing. Let me just show what should be available. GenBank accessions, not clade, actually, that's only for specific viruses. Collection year, geographic region that it was collected from, host and common name, isolation country, lineage, product, species, strain. Those fields should be available to you, and we'll try to solve that and make them available to you. There's a metadata table that has those columns in there, and you should be able to see them in there, which might be helpful to you. So with that, I'm going to conclude viewing the tree.
We have two Zoom questions. The first question is, which model would be more useful for viral analysis? Would you also use LG?
Could you make the same alignment and then run various RAXML trees with the different models looking for the best likelihood score?
You certainly could. You certainly could.
Because I think that's what I would do, although I don't know if that is the right way to do it.
RAXML actually has a run mode where it will search through its available models and find the optimal model. And we should make that available. That is available in the codon trees version of tree building, but we need to enable that in this version here too.
The second question is, for some of the sequences, the bootstrap number is 4. What could be the reason for it?
One obvious reason is if the branch lengths are very short, as they are in this clade here. I think these are Buchner's. Here's a 0. There's a 4. Those are very poorly supported, but it's not surprising that they're poorly supported because this tree shows that there's approximately 0 amino acid difference between them because their branch lengths on the tree are close to 0. You can zoom in on the x-axis using this tool, and you can move the tree. So even when you zoom in farther, there's still not little stubby branches there. It looks like it's approximately absolutely 0. So I'm surprised that there's even that. The answer is there's very little difference between those sequences. They're almost identical. So it's not surprising that you can't be confident on the relative relationships among them. Also in this clade too, they're very close.
But to me, the resonating fact of that is that there's 100 bootstrap support separating the upper from the lower ones.
Yeah.
So I feel confident that those are different. Upper is different than lower.
Yeah, when these branches are longer, as most of the branches are on this tree, they're accompanied by 100% bootstrap support. So it's just a lesson in interpreting trees. When they're nearly identical sequences, it's just not surprising that you can't say which one's more closely related to the other within the group of nearly identical sequences.
I haven't used Archaeopteryx that much. And big tree, I could switch it around at the base. Like if I wanted that lower clade to be on top and the upper clade to be on the bottom, I could click at the 100 node and flip it. I don't think you can do that in Archaeopteryx, can you?
Yes, you can. You can,
Oh,
for
Well,
example,
then it's much more awesome than I was allowing myself to believe.
there. And look at it, it even animates the transition. You have to click on the node and swap descendants is the function.
Oh, that's pretty awesome. I often do this kind of thing when I'm writing a publication. I like to do the established order that most of the people like to see the tree, with certain clades at top. So this allows me to make my tree look like their trees.
This O button applies that ordering to make the densest branches cluster at the top, and I guess clustering at the other, clicking it again, flips that to the opposite. Also, there's these 3 views of the tree here, and this one simply aligns the tip labels off to the right side. That can sometimes make it easier to read the tip label, so you can maybe, sometimes it's more difficult if the tree is very uneven and they're very different depths. And then there's this clade view where the branches are not shown proportional to the amount of changes, but just to a fixed unit branch based on how deep the tree is. And that can be good for understanding the relationships that are suggested by the tree, even in the cases of where the branch support is 0 as here and 4 as there. Even though there's no support for it, you can at least resolve what the tree topology is.
Oh, and if I was in that view, because this is something that often confounds me, especially when the branch lengths are very short, could I rearrange the order using this where I can actually capture those branches and then have it preserve that when it goes back to the phylogram, which is what I actually want?
You mean you want to switch them like
Yeah,
this?
let's say I wanted to switch that. And then I want it to look like that. Now click phylogram. And if it's the same,
Yes,
oh,
it kept that tree.
okay. Any mean thing I have ever said about this, I take it back, because that's something I really like. But I have never said a mean thing about any of our tools. You must know.
You can adjust the size of the labels. I've been dealing with large bacterial trees where there's more labels than you can see written. In fact, if I make these really big, it's got a feature where it's called Dynahide, where if they're so big that they're going to overlap each other, it will hide every nth label so that they don't write over each other, and you can turn that on and off. There's a lot of features on branch width. You can make the branches thicker. And let's see, one other thing is you can download a PNG or an SVG or NUIC or PhyloXML format. So there's a lot of functionality. The one thing I'm very unhappy about is that we don't have the species and strain identifiers as labels here in this tree, but we're going to make sure we fix that. I consider that a bug and thought this was fixed a while ago, but it's still haunting us.
One additional question. Under what circumstances would you use gene tree versus phylogenetic tree?
For bacteria, if you really want to know the relationships of them as species, I would just go straight to a phylogenetic tree with a codon tree program behind it, because it will concatenate multiple protein alignments and 10 or 100. I think 100 genes is a really great amount of data to build a tree from. That takes so long though, but you're right. If you really want to resolve fine relationships at the species level, the whole genome phylogenetic tree approach is much better. This is if you're interested in the particular gene. If you want to know the relationships among copies of a gene, which could include duplications within species, and you could be interested in tracking horizontal transfer, you might want to compare the topology of the gene tree to the species tree that you've built for the same exact genomes.
I see that in a lot of papers where they're exploring a particular gene and then want to look at the greater phylogeny so that they can see if it's that within this gene, it's evolving at a different or more rapid rate than the species as a whole.
One thing I'll say is that we also have, here's the alignment, this top file that I clicked on. This MSA viewing tool will view it in the other window. And just as I said that it trimmed off that ragged end, that trimming function. Here's the leftmost column of the alignment and it's been trimmed in. There's a report actually that describes how much trimming went on, and that's in this HTML report, and you can view it here. It's got this view of the tree, analysis steps, align with MAFT, trim alignment, ends trimmed, 16 columns on the left, 6 columns on the right, just to hit that functionality. That, I think, is all that I have on this topic, and I apologize for the labels not being all available. We're going to fix that. And then the other thing is enabling auto detection of the optimal protein substitution model. We should definitely enable that, and I'll make sure that that happens. On the nucleotide side, I would say just go with the GTR model, generalized time reversible model, and I don't think you can do better than that. The question is, can you highlight a particular node? And yes. What if we click this? That's a much better view of the tree. Suppose you are interested in this whole group here, and you need to use the mouse, and you need to be pretty close to the juncture of the horizontal bar with the vertical bar to get mousing over the node. And you can tell because it changes the icon to the pointing finger icon. You can select the node. There's a red dot for that node, but maybe more usefully, you can select all of the descendant nodes, all of the external nodes descended from that node. And I'm glad you brought that up. Thank you. This saves a bit of face, I have to say, because having selected that, the green bar gives you functionality to look at either the features data or genomes data on that selection. And genomes data will tell us the species and strain. And so let's just click on that. It's going to take us to a genomes list for those tips. And thankfully, we can see that they're Blackmonia and Wigglesworthia and more Blackmonia.
Blackmonia are ant endosymbionts. I sort of cut them out from this workshop, but they were so insistent they wanted to be included again, apparently, so they invaded Alan's subconscious.
Well, this genome group is theta reduction from all these endosymbionts. These are in the gamma proteobacteria, I believe. I feel really good that there is a way for you to at least temporarily be able to understand what the species are for different regions of the tree. I'm going to deselect those. I want to select this node and see what those are. Those are Buchneras. There is actually a view that shows the desired labels on this tree. Just you have to open it up in a different way. If you go down to the phylo-XML tree generated by the gene tree output, and don't click the blue eyeball at the top, click the view icon in the green bar, And at first it looks ghastly, but you do notice that the species name is in that list. And now if you turn off the node name, the species and strain name are available. It still looks ghastly, but you can fix that by clicking the X plus button, which grows the X-axis. And now, in fact, it actually looks like a real tree. And Evan from Michigan State University, he deserves, probably doesn't want, but he deserves a bunch of chocolates for that. And now you can turn strain off, strain on. In fact, I like it with strain off because the strain identifiers don't mean anything to me. Let's try adding host common name. Some of them actually have it. Green peach aphid. Yeah, that's the only example of host common name being available. We turn off the species too. It's very explicit which ones actually have that field. Let's try collection year. Some of them have collection year. This is the level of being able to turn on and off the metadata about the tips that I think makes this tree viewer really useful. I just was blanking on the different ways of opening the tree tool, and that big blue eyeball at the top, while enticingly easy to click on, I guess is not the best route entering the viewer for this tree.